Hello students, today we are going to discuss reproduction in eubacteria. In case of eubacteria or true bacteria, the reproduction is mainly asexual. Which we know is dependent on single individual. It means it is uniparental, in which there is no gamete formation and no fertilization. A primitive type of sexual reproduction is also reported, in which there is no gamete formation, no fertilization, but genetic recombination is there. Although the true type of sexual reproduction is absent, but a primitive type of sexual reproduction, which is known as parasexuality, is present in bacteria where there is no gamete formation, no fertilization, but genetic recombination is reported through some special methods which we will discuss later. These are transduction, conjugation and transformation. In asexual mode, we have mainly two types. That depends on the environmental condition in which that bacteria is present. It can be binary fission or it can be spore. Binary fission takes place when the environmental condition is favorable, and spore formation while unfavorable environmental condition. Now, what does that mean? Favorable conditions means there is sufficient nutrients in the environment. The temperature and moisture content should be favorable for the growth of bacteria. Now, let's discuss each mode individually. First of all, we are going to discuss about the binary fission. The single bacteria divides to form two bacteria, and those two bacteria will divide again to form two each. It means in the next generation in the first generation we'll get two bacteria, and in the next generation we are going to get four bacteria. So the growth is geometrical. It means one bacteria will divide to form two, and these two bacteria will divide to form four bacteria. So this is the first generation and this is the second generation, and in the third generation we'll get 8 bacteria. So, if the question is asked in the examination that to get 8 bacteria, how many generations are required? So, it will come 3 generations. There is a general formula to calculate that, and it is 2 to the power n is equal to x. It means the bacteria needs to divide for n number of generations to get x number of bacteria. Now try solving one question that is oftenly asked in competitive examinations. The question, such type of questions are asked in the examination. If bacteria divides every minute and takes 15 minutes to fill the 1/4 of a cup, how much time will it take to fill the cup completely? In this question, it is given that bacteria divides every minute. It means if the cup contains, if this is the half and this is 1/4, and in 15 minutes 1/4 is filled, so it will take another minute to complete the half of the cup, to fill the half of the cup, and in the next minute it will fill the cup completely. So finally we get 15 plus 1 plus 1, that is 17 minutes. Now let's see the process of binary fission. We all know the structure of the bacterial cell. This is the cell wall. We know that cell wall is made up of peptidoglycan, and inner to the cell wall there is the membrane which shows certain outgrowths and certain ingrowths. This is the plasma membrane. And we know the nucleoid, that is the genetic material of the bacteria, is present attached to this ingrowth of plasma membrane, which is known as mesosome. This is the nuclear material and it is known as nucleoid or incipient nucleus. Now let's see the binary fission process. It takes place in 3 steps. So what are the steps? First, DNA replication. Second step is septum formation in mesosome. And the third step is cross wall formation. We know that DNA in bacteria is circular. So after the replication, there are two rings. So while that replication process, those two rings are attached at some point to appear like the theta symbol of the mathematics. So it is known as the theta model. It was given by Cairns, so it is known as Cairns theta model. The DNA material gets replicated, then a septum appears in the mesosome at the point of attachment of DNA with the mesosome. So this septum is formed in the mesosome. In the next step, an invagination appears in the plasma membrane, which deepens to complete the division of the cell, and the two cells get separated. This mesosome is shifted towards the middle location of the daughter cells, future daughter cells, and this is the way the binary fission gets completed. During unfavorable condition, spore formation takes place. The spores formed during unfavorable condition is known as endospore. Inside the bacterial cell, the complete protoplast comes at the center and secretes a thick wall around it. This thick-walled structure is known as endospore. It is released outside. It is very tough and resistant to temperature, desiccation and chemicals. That's why it can survive for years. It is a thick-walled structure which is resistant to temperature. It can withstand temperature from minus 100 degrees C to plus 100 degrees C. It is also resistant to temperature, chemical and desiccation. Let's see the structure of endospore in detail. On onset of favorable condition, we have already discussed that spore formation is there during unfavorable condition, and on onset of favorable condition, these endospore will germinate to form the new individual. Now let's see the structure of the endospore, which makes it resistant to harsh conditions. This is the cytoplasm, which contains the nucleus. Around the cytoplasm, a layer is present which is known as, this is the nucleus. the cytoplasm. Outer to the cytoplasm, a layer is present which is known as core wall. Around the core wall, again a layer is present which is known as cortex. It also has the cytoplasm with different density than the cytoplasm present at the core. This layer is known as cortex, which contains an anticoagulant known as calcium dipicolinic acid. It is an anticoagulant which makes it resistant to temperature, chemicals and desiccation. Around the cortex, a different layer is present which is known as spore coat. Around the spore coat, again a layer is present which is known as exosporium. Due to this thick wall, it can survive through the harsh conditions like temperature from -100 degrees C to +100 degrees C, the presence of some harsh chemicals in the environment, and desiccation, that is unavailability of water. On onset of favorable conditions, these spores behaves like seeds These endospores behaves like seeds, and on onset of favorable condition, it germinates to form new individual. This was about the asexual reproduction. Now come to the next topic, that is parasexuality in bacteria, which is a primitive type of sexual reproduction present in bacteria. So as we have discussed earlier, there is no gamete formation, there is no fertilization, but genetic recombination is present, which is through some special methods like transformation, conjugation and transduction. Let's start with transformation. The transformation was discovered by Griffith. His experimental material was Streptococcus, or Diplococcus, in transformation, the cell, there is no direct contact between two bacteria. The bacteria absorbs the DNA present in the environment and gets transformed. That's why it is known as transformation. So let's see the Griffith's experiment. The experimental material is Diplococcus, and he took mice and introduced the strains of that Diplococcus inside the mice. There are two strains. One is R type, that is the rough strain of the bacterium Diplococcus, and S type, that is the smooth type. The rough type or rough strain of that bacterium is avirulent. Avirulent means it is non-pathogenic. It doesn't have the ability to cause any disease. It is non-pathogenic. And a smooth type has ability to cause the disease. It is known as virulent type. Virulent or pathogenic. Now he introduced in the first step, this is the step 1. In first step, inside the mice, he introduced the R strain or R type, and the mice survived. In the next step, he introduced S strain of the bacteria inside the mice, and the mice died. In the third step, he killed the S strain with applying the heat. So heat killed S strain was introduced in the mice, and the mice died. The mice survived. To his surprise, in the last step, he combined first and third step in which the mice survived. But when he combined these two steps, the mice died. So he introduced living R strain plus heat killed S strain, and the mice died. When he examined the fluid inside the mice, he found some living S strain which he did not introduce inside the mice. So he gave one hypothesis that inside the mice, the living R type got converted to S type due to presence of something that came out of heat killed S type. So he named it transformant. According to him, this transformant came from heat killed S type, which converted the living R type to S type. But he did not explain the chemical nature of this transformant. After few years, it was proved to be DNA by the work of scientists. Three scientists were there: Avery, McCarthy and MacLeod proved it as DNA. This was about the transformation. Now come to the next process known as conjugation. Let's revise this experiment once again. What is transformation? That is absorption of the DNA from the surrounding medium and getting transformed to some other form. Here the Griffith took mice and a bacterium, Diplococcus or Pneumococcus or Streptococcus, and he took two strains of that bacteria, one R type or rough type, which is avirulent, which doesn't have ability to produce the disease, and one S type or a smooth type, which is virulent, which can be pathogenic to the mice. What he did? He introduced, in first step, he introduced the living R type inside the mice and the mice survived. In the next step, he introduced S type and the mice died. In the third step, he killed the S type by applying the heat, so it lost its ability to cause disease, so the mice survived. But when he combined the living R type with the heat killed S type, the mice died. When he examined the fluid, the pleural fluid from the mice, he found some living S type which he did not introduce. So he gave the hypothesis that something came out of that heat killed S type which transformed the R type to S type. He could not tell the chemical nature of that transformant, but later by three of the scientists, Avery, McCarthy and MacLeod, this transformant was proved to be the DNA. Now come to the next special method of genetic recombination, and that is conjugation. Conjugation was discovered by Lederberg and Tatum in E. coli. In transformation, there was no physical contact between the bacteria. Here, there is the physical contact between the bacteria. We have seen that the plasma membrane has certain outgrowths and certain ingrowths. We have studied about the ingrowth. These are mesosome, and these outgrowths are called pili or fimbriae. Pili or fimbriae. The pili helps in formation of conjugation tube when it comes in contact with the bacteria. So we know that along with for the simplification, I am here representing the genome of the bacteria as a circular double-stranded DNA. This is the bacterial genome. Along with the bacterial genome, it contains an extra chromosomal DNA which is known as plasmid. This is plasmid. It is also known as extra chromosomal DNA or mini chromosome. The bacteria which has the plasmid is known as F plus bacteria or male bacteria or donor bacteria. The bacteria which does not have the plasmid is known as F minus or recipient or female bacteria. It is F minus or recipient F minus or recipient or female bacteria. The plasmid contains the gene for conjugation tube formation. So it is also known as fertility factor. The bacteria which has the plasmid can form the conjugation tube. This is the conjugation tube. This plasmid is auto-replicative. It has the ability to form its own copy and transfer it to the recipient bacteria. In this process, the F plus bacteria combines with F minus bacteria to form both F plus bacteria. But there is no genetic recombination here. Sometimes what happens, this plasmid gets associated with the genome of the bacteria, and now it forms a copy of it which also contains the part of the bacterial genome. The whole genome cannot get transferred to the recipient bacteria because this conjugation tube is formed only for 1 or 2 minutes, while it takes around 90 minutes for the complete genome of the bacteria to get transferred to the recipient bacteria. So a part of it comes to the recipient bacteria. Now this part contains a part of the genome of the F plus bacteria. So it has the allele for the gene present here. So there is possibility of crossing over or genetic mixing. The part which comes from the F plus bacteria is known as exogenote, and the part which is there in the recipient bacteria is known as endogenote. Together, these are known as merozygote. This is conjugation. Now there is genetic mixing. So let's discuss it again. Conjugation is by physical contact between two bacteria. It was discovered by Lederberg and Tatum in E. coli. In this process, the bacteria, F plus bacteria with plasmid comes in contact with the recipient bacteria. The plasmid is associated here with the bacterial genome, and this bacterial genome, when there is association of the plasmid here, is known as episome. So bacterial genome plus plasmid is called episome. The bacteria with episome is known as HFR bacteria. HFR bacteria is high frequency recombination bacteria. When this HFR bacteria comes in contact with the recipient bacteria, it transfers a segment of the bacterial genome to the recipient bacteria, which contains the allele present in this bacterial genome, and that's why here crossing over is possible. The genetic part which comes from F plus bacteria is known as exogenote, whereas the part which is there inside the recipient bacteria is known as endogenote. Together, exogenote and endogenote are known as merozygote. So this was about the conjugation. Now come to the next process, known as transduction. The transduction is genetic recombination with the help of virus. It is genetic recombination with the help of virus. This virus which attacks on bacteria are known as bacteriophage. Virus attacking the bacteria is known as bacteriophage. Let's see the structure of the bacteriophage. It is made up of protein and nucleic acid, that is double-stranded DNA. This structure is known as head or capsid of the bacteria, which is made up of protein. This is the tail. At the end of the tail, an enzyme is present which degrades the bacterial cell wall. This enzyme is lysozyme. These are the tail fibers. The nucleic acid present here is double-stranded DNA. When this virus infects the bacteria, it brings transduction. In stepwise process, I'll explain how does it help in genetic recombination. The transduction was discovered by Lederberg and Zinder. The bacteriophage infects the bacteria the virus scans for some special receptors where it can get attached. After being attached on the bacterial cell wall, with the help of lysozyme, it degrades the bacterial cell wall, which is made up of peptidoglycan, and it squeezes its nucleic acid inside the bacterial cell. So this comes inside the bacterial cell. The bacterial cell has its own genome, and this one is the viral genome. This is the genome of the bacteria. And this part is viral genome. Like episome, this viral genome gets associated with the bacterial genome. In the next step, this bacterial genome and viral genome gets integrated. Till the time the viral genome is integrated with the bacterial genome, it is not harmful for the bacteria. And in this state, the bacteria undergoes binary fission and the viral genome is replicated with the bacteria. This type of cycle is known as lysogenic cycle. Till the time viral genome is associated with the bacterial genome, it is not going to harm the bacteria. But sometimes due to asymmetrical accession, a part of the viral genome takes control of the complete enzymatic machinery of the bacteria and degrades the rest of the genome part. It degrades it and starts synthesizing its own part like capsid, nucleic acid and tail fibers. Next step is packaging of these bacteriophages. Packaging of virus. These viruses here there is assembly of all the components to form the bacteriophage along with its genome, which contains a part of bacterial genome as well. When packaging is done, it lyses the bacterial cell and releases these bacteriophages, which are the real transducing viruses. Now, when these viruses, which we call bacteriophage, infects the fresh bacteria, it also contains a part of the previous bacterial genome which has the allele of the gene present in the bacterium, which brings genetic recombination. So this is about transduction. Let's revise it. Transduction was discovered by Lederberg and Zinder in bacterium Salmonella. The virus which infects the bacteria is known as bacteriophages, which has two parts, head and tail. Head is made up of protein and it contains the nucleic acid, which is double-stranded DNA here. When this bacteriophage infects the bacteria, it introduces its nucleic acid inside the bacteria, which gets integrated with the bacterial genome, sometimes due to asymmetrical excision. A part of the viral genome with bacterial genome takes control of the complete enzymatic machinery of the bacteria and starts synthesizing its own components. After the components are synthesized, it gets packaged to form the new virus inside the bacteria. Once this packaging is done, it degrades the cell wall of the bacteria and releases these viruses. Here there is lysis of the cell or lysis of the bacteria. It is known as lytic cycle. Till the time the viral component is associated with the bacterial component, it is not harmful for the bacteria. But here, when the new bacterium is formed inside the bacteriophages are formed inside the bacteria, it lyses the bacterial cell, destroys it and comes out, which contains the part of the bacterial genome also. Now these are the real transducing viruses which are carrying a part of the previous bacterium to the fresh bacterium where genetic recombination is possible. So these were the methods through which the genetic recombinations were possible inside the bacteria. As we have seen that there is no gamete formation, there is no fertilization, but genetic recombination is possible. It is named as parasexuality, which is considered as very basic type of sexual reproduction.